the Pediatric Lounge, a podcast taking you behind the door of the physician's lounge to get a deeper insight into just what docs are talking about today. From the clinically profound to the wonderfully routine and everything in between. Hi, George. It's a Friday and we have a phenomenal podcast today with three endocrinologists to talk about the history of insulin and more.
Yeah, today we have Dr. Latif, Dr. Haller, and Dr. Kaiserman. And the topic of discussion is going to be from starvation diets to insulin, 100 years of innovation as we run together towards a cure.
Today we're honored to have three distinguished professionals joining us. First, we welcome my friend, Dr. Kashif Latif from Memphis. He's an internal medicine diabetologist, but he sees children's, whose expertise brings valuable insights to our pediatric community. We're also joined by Dr. Michael James Haller, a professor at the University of Florida in Gainesville, who brings extensive clinical experience to today's discussion. And finally, we have Dr. Kevin Kaiserman, who serves as Senior Vice President of Medical Affairs and Therapeutic Area Head for Endocrine Disease and Mankind, bringing a unique perspective to medicine from the pharmaceutical industry. As a pediatric endocrinologist, he has helped more people than he could reach in the clinic alone at one time. Welcome, gentlemen.
Thank you. Thank you.
Before we get started, just a brief note to our listeners. I invite you to share this episode with all your friends and colleagues. A lot is happening in T1 space, especially in early screening and universal screening. A resolution is before the American Academy of Pediatrics Leadership Council. A submission has been made to the U.S. Preventive Service Task Force, and a Minnow Fellowship led by Barbara Davis Center will be available free of charge to all pediatricians the Thursday before CME at NCE in Denver, Colorado this year. There are new forms to deliver insulin and monitor insulin today. We will be discussing experimental and non-branded name FDA-approved and non-approved interventions. If an FDA package insert exists and the name comes up in the conversation, we will include it in the show notes for you. Please consult with your diabetologist before considering any treatment. Dr. Latif, why not pediatrics?
I thank you so much, sir. It's a pleasure to be here and share my experience. Was an internist at the outset when I started my medical career. Events led to the state where my 11 months old son developed type 1 diabetes in 1997. Then after I went back to train to be an endocrinologist and worked heavily with children and was trained directly by one of my professors from the Bonner Children's Hospital. So did not do a formal pediatric fellowship, but had been seeing children with diabetes from the outset of my career.
Wow. And how's your son doing today?
He is doing fantastic. He is 29 and a half now, and he is a software engineer, works for Amazon, wears an automated insulin delivery system from Medtronic, and he's doing very well.
So a long way from when you had to give long-acting insulin twice a day, measure it, and poke the finger to see where he was at?
100%. And we're going to talk about evolution, and I can speak from my experience. Then in 1997, the insulins were not what we have today. We had Lantus, Ultralente, regular insulin. We did not have any of the faster-acting insulins that we use at this point. And just for historical comment, when I started injecting our son with insulin, we used the NPH, and it was a long-acting insulin that was being injected twice a day. And at his age, he was on a minuscule dose of 1 unit in the morning.
Dr. Haller, why did you become a pediatrician and then a diabetologist or endocrinologist?
I've always just preferred being around kids than adults. They are brutally honest and always fun to be with. So I knew I was going to be a pediatrician once I was dedicated to medicine. My grandfather lived with type 1 diabetes, so that was part of my interest. But then during medical school, I had incredible mentorship from Janet Silverstein and Des Schatz and Mark Atkinson, who are known in the pediatric endocrine world, who really steered me to a career in type 1 diabetes specifically. And now I've been working in that space for the last 25 years.
Well, Dr. Kaiserman, why pediatrics? And what is your connection with autoimmune insulitis or type 1?
Well, good day, and thank you for including me in this wonderful panel. For me, my journey in working with people with diabetes started about 30 years ago. And just by chance, my career started with the publication of the Diabetes Control and Complications trial, which really demonstrated the connection between the hemoglobin A1C levels and the risk for diabetes complications. And so I was able to start my career at a time when there was a lot of hope about helping people living with diabetes to fulfill their lives, reach their goals, and really led to the explosion that we've seen in the last 30 years of new diabetes therapies and technologies that we'll be discussing further today.
Thank you. And Dr. Haller, you've done some phenomenal studies, but a couple of them caught my eye because it's just interesting to me. And one of them was a study using the EHR to look at the prevalence of T1D. Why do you think that's an important thing to do, is using that tool?
As we get further and further into the age of AI, we really have to use those tools to make sure that we capture everyone who's out there living with type 1. And importantly, as you started off the podcast with, we now have the opportunity to screen and identify folks with early stage disease. And doing that in a directed way makes sense. And so using EHR data to identify folks with type 1, whose family members may or not have been screened, is a low-hanging fruit. Ultimately, we hope to have enough data and interventions to support more broad general population screening. But even now, we don't do a thorough job of identifying everyone. So we can use the EHR to get those folks and also to correct misdiagnoses we see quite often in the adult world. I'm sure Dr. Latif can speak to this. Many folks with type 1 incorrectly diagnosed as initially having type 2. And you can really use EHR data to actually help you make sure you're sorting those patients and identifying them correctly.
So that alludes to another wonderful paper that your group wrote, right, which is actually identifying an algorithm to query the EHR. I'm guessing, so I'm not a diabetologist, so I'm guessing that if you just type in the ICD-10 code for type 1 diabetes, you get a lot of noise, a lot of errors. So you identify a way to have a built-in process to identify them off of an EHR.
Yeah, that's right. Unfortunately, most of those codes were really designed just for billing. And so trying to use them to correctly identify a patient's type of diabetes is certainly problematic. But it's not terribly challenging to build algorithms for systems to go through and really sort patients based on what's in their chart and the kind of diabetes they most likely have, given their weight and their history of insulin use, family history, and those sorts of things. So this can be used to really correct your list and make sure that you're starting with a group of people more likely than not really do have type 1 diabetes.
That's a phenomenal paper. Now, you have published several papers on Project Echo, and that's one fascination that I have. What is Project Echo? Why is it important? Why are you using it as a pediatric endocrinologist/diabetologist?
Yeah, thanks for asking. Project Echo stands for Extensions of Community Health Outcomes. And it was a concept really that was first developed by Dr. Sanj Arora in New Mexico. He's a gastroenterologist, and he was interested in trying to teach people how to effectively eradicate hepatitis C, which we can do now. But there aren't enough gastroenterologists who are experts in hep C to go around. And the reality is most of these patients are managed by their primary care doctors. And so he set out to set up a network of primary care offices who would participate in an all teach, all learn web-based weekly or monthly seminar series that would include case reports coming from the actual docs. And through that mechanism, they would learn how to provide subspecialty light care, more than the primary care average doc could do, not quite as intensive as a subspecialist, but certainly enough to teach very specific care like that. And that model has now been applied to hundreds of different diagnoses all over the world. Our group was fortunate to get some generous funding from the Helmsley Charitable Trust to do the first echo diabetes program in collaboration with Stanford University. And we did that not only as a clinical entity, but as a trial to really try to study for the first time if echo had impact, not just on the physicians feeling more comfortable in providing subspecialty care to people with diabetes in this instance, but did it actually impact patient outcomes? And in fact, we did show that the echo diabetes program not only improved the primary care doc's comfort level with prescribing CGMs and giving insulin, but it actually did have an effect on improving A1C and reducing hospitalizations and patients out there living with diabetes, many of whom don't have access to endocrinologists because there's just so few of us and too many patients that need to be seen.
Do you think that model is ripe for using to diagnose early stage 1, stage 2 autoantibodies in the primary care arena?
I do. In fact, we've proposed exactly that to a number of funders and hoping to have some opportunities to do that in Florida specifically over the next year. It's a fairly easy concept to teach, certainly pediatricians, and I think family practice and internists, why to screen for type 1 diabetes antibodies, who to screen for those autoantibodies, and how to initiate follow-up. We actually did that as part of our echo diabetes program. That was one of the modules we covered. We were able to get kits for screening placed in a number of federally qualified health centers. And these are places that take care of patients who are significantly underrepresented in clinical screening and research in general, and show that even in those challenging places, we could get patients and families screened for type 1 antibodies. And we also use that opportunity to correct misdiagnoses. We had many echo centers bring us their cases of folks they thought were likely type 1, and we were able to teach them how to appropriately test those patients for antibodies and confirm their suspicions and augment their treatment because of it.
Wow. I just think that's a fascinating field because there's not enough peds or adult endos. Most counties don't have an endocrinologist. And although the data doesn't bear this out in the clinic, type 2 diabetes is becoming more frequent than type 1 in pediatrics because of the obesity epidemic. So you see all these dysglycemic children who are obese. And there's just no way that you can have the endocrinologist see them all. And you're going to have to, in the primary care arena, at least manage them, whatever you can do for them, and prevent disease progression and diagnose them properly and hand off those that are just too complex for me to take care of. But it's a wonderful project. I have a question because you were the lead author on the ISPAT guidelines for diabetes care and screening. And the only thing that was missing is you didn't have a 1 positive antibody dysglycemic pathway. And that's the most common thing I have in the clinic. And I just go back and do their hemoglobin A1C every 3 months and repeat their antibodies once a year for 3 years. But I feel like I'm skating in thin ice because I'm not a diabetologist.
Yeah, thanks for pointing out that omission. I think that's because it's an area that we really don't have a robust enough data set to provide strong recommendations for. The majority of folks with a single antibody who have dysglycemia are not going to be patients who ultimately develop type 1. They may have phenotype that's more like a type 2 patient if they do progress to dysglycemia or frank hyperglycemia. But there's still 3 to 4% of those per year that do progress and do look like type 1. And they unfortunately just don't fit our standard of defining stage 1 diabetes that has having 2 autoantibodies. So you're right. And that is an area that we can continue to work on. I think you're doing your patients a good service by making sure they don't just get ignored. But I don't think all those patients need to get referred to endocrinologists, certainly not to try and consider treatment with immunotherapy or anything like that at this point, because we really don't have a strong enough evidence base for that.
So just watching them is okay.
I think that's reasonable. Certainly repeating the antibodies, making sure they really are just single autoantibody positive and don't seroconvert to being multiple antibody positive. Once you're over 12, 15 years of age, if you've got 2 autoantibody tests, 2 separate tests, and they show you only have a single antibody, very unlikely to progress to developing a second antibody. Now, that doesn't guarantee you won't progress to developing diabetes, but you're really unlikely to seroconvert.
Great. Thank you. Dr. Kaiserman, why did you transition from my beautiful home state back to California and from the clinic to the boardroom?
It was a wonderful journey. So I have to say that 30 years of working with children, adolescents, and young adults with diabetes has been a really fulfilling career. I trained at UCLA in both pediatrics and pediatric endocrinology, and then stayed on faculty there for a number of years, leading their diabetes program. Moved across town to Children's Hospital, Los Angeles, and worked with their diabetes program for a number of years after that. And then went into private practice and had a really wonderful career working directly with people living with diabetes, doing lectures around the world as well as clinical research. And that was all very rewarding. And then an opportunity arose for me to join industry. And as you talked about earlier, to bring potentially new therapeutics to people living with chronic illnesses is an opportunity to really reach a lot of people that you otherwise would never be able to interact with. So I think all are really important aspects of care. I've enjoyed every aspect of my career, and they're all a little bit different, but it's just been a wonderful journey, and I'm grateful to have had it.
Wonderful. And then I don't know who wants to take this question, but this just blew my mind, right? Because it's hard to have perspective. I started residency in 89, and I did use NPH and Lente and regular. That was all that was available then. And when I was listening to a podcast about Novo Nordisk, they mentioned that in the 1920s, there is one therapy for diabetes, starvation diet. What was that?
I can jump in, and my colleagues are happy to have them add. But prior to the discovery of insulin, and the first patient was treated in January of 1922, a young teenage boy named Leonard Thompson. And prior to that, without having a treatment for diabetes, the treatment at the time was a very restrictive diet, very high in fat and very low in carbohydrate, as a way to try to keep people to prolong life in that time. And the important thing to think about here is that some of those patients who were treated with that therapy actually survived to a time when insulin was discovered. And Frederick Banting and MacLeod from the University of Toronto shared the Nobel Prize in 1923 for the discovery of insulin. So it's a pretty amazing journey, and certainly there's a lot of wonderful documentation written about this journey of the discovery of insulin and how it really has impacted so many lives around the globe.
So it was very interesting. This came out of Toronto, right? And what they did is they grinded dog's pancreas, and then from there they got the insulin, and then they gave it to a human person, and it worked. But they had no way of measuring glucose in the serum at that time. So it was just see what happens. And then they gave a patent to Eli Lilly. And this is the second time I come across a pharmaceutical house doing something to me strange. The first one was Merck. When they were doing the measles vaccine, they need eggs, but they need absolutely clean eggs. So they became the largest poultry company in the US because they wanted the chickens that could not get leukemia and produce the perfect egg for them to use to create the vaccine. And then Eli Lilly was able to commercialize insulin in the US by grinding mostly beef pancreas. And they bought the beef pancreas from cattle ranchers all over the Midwest. And that's how they produced insulin, which is amazing history, right? We don't think about these things. And apparently it was a very primitive process. They just would grind the pancreas of the cow, then throw bleach on it, and then there you go. And it wasn't even an injectable, it was in a pill. And people had to boil water, dissolve the pill, and then give themselves shots. And it was very short life. So you had to give yourself a lot of shots to maintain life. And then I think Dr. Latif knows his story. Out in California in 1970, someone came up called Genentech. And what happened then?
Yeah, then the yeast evolved, and we have recombinant insulin using E. coli and then a lot of nature's products out there. And so the initial insulin, as I will go back, the history would be that I used on my child was NPH, and small doses, 1 unit in the morning, half of one at night, did not have the proper tools to inject. And thereafter, we started having in the 2000s, we started getting the rapid insulin analogs. And now we are on the way up to an inhaled formulation. So lots of evolution. And I think Mike and Kevin can focus walk us through.
So let me ask you for the non-endocrinologist. When I was in medical school, and I tell this story just because you have to be humble. I wrote a prescription for insulin for an adult. And the professor, my mentor, whoever does the attending, said to me, "What are you going to ask the patient?" I'm like, "He's got diabetes. He needs insulin. What else am I supposed to ask him?" And he's, "Oh my God." He goes, "Please ask him if he has a refrigerator." I'm like, "Why would I ask him that?" He goes, "Insulin needs to be refrigerated. If you give him that prescription and he doesn't refrigerate the insulin, it's worthless. You're wasting his time, wasting money. Go ask him if he has a refrigerator." So I was preparing for the podcast. I saw that 1990s Novo introduced insulin pens. So was that the first time that insulin didn't need to be refrigerated?
Then all insulin is more stable the cooler you keep it for longer because it's a relatively short peptide. So it's always recommended to keep it refrigerated for long-term use. Once it's out of the refrigerator and opened, a single vial or a pen, they generally have a half-life or a shelf life, a usable life of a month. But even that's really arbitrary. That's what the FDA has allowed. Our group has actually studied this because we have a particular interest in many areas in the world where insulin is still scarce and people die from lack of access. It even happens in the US, unfortunately. But the reality is insulin is actually pretty stable. If you don't get it extremely hot and you don't freeze it, it does continue to lose potency. But even insulin out of the fridge, sitting at room temperature up into the low 90s, actually maintains about 90% of its stability over about a year. So while we certainly do recommend keeping insulin cold, I think we probably worry about that more than we need to nowadays with the insulins that we have available to us.
So how does that work when you have an insulin pump like in DC? It'll get 95 degrees outside and 90% humidity. And in that pump, it doesn't have refrigeration. So it's still stable at that kind of...
Exactly. So reminding your patients that they're only wearing that single insulin pump with that single insulin cartridge for 2 or 3 days at a time. So they're replenishing it with insulin that hopefully has been kept in the refrigerator. So during those just few days that they're using that one pump or one cartridge, the insulin doesn't degrade enough to cause problems.
Well, and Dr. Latif, how much did the insulin pumps and the CGMs change your life as a parent?
From the time of gauging what the symptom may be of an infant, and there was no communication, "Hey, I'm not feeling well," all the way to seeing the glucose real time fluctuate and to adjust therapy, it has been the life changer. It's a blessing in disguise. It's a life-saving tool that has been developed, and it has had its evolution over time. And we are now at a point where we have 4 and 5 systems, which are hybrid closed loop systems where the delivery of insulin is for the most part automated. From guessing and delivering, giving life-saving therapy to the point where we know exactly how much to do and what the result is, it has been absolutely phenomenal.
And how much does it add? Because I saw that there is a new meter or CGM that will also measure ketones. How much does that add to the management?
That is going to be something that we'll see in evolution, because the ketone meter is going to give us the flip side of what the ketones are doing, and that will help us guide therapy. But I think there's so much to learn with the ketone production and disposition as it relates to real-time changes in glucose levels. So really interesting field, and Abbott has one developed already. It's not approved for use in US at this point in time. Probably have it in Europe before it comes to US, but there's going to be a lot more to learn. Right now, we just do ketone testing at a point of care, like at one time, to evaluate if somebody is going into ketosis, but to see it moving in conjunction with glucose levels, I think we're going to have a lot of learning. And I'm sure Mike and Kevin can add to that.
Dr. Haller, so after all this, so I'm still stuck in NPH, and that's all I got. I got to prick the kid before every meal and have a sliding scale to give him some regular insulin. That's my care of a child living with diabetes. Now I understand that there's inhaled insulin. We can do without the shots, and why, and how fast does it work?
Yeah, thanks for asking about that. Yeah, the field was forever changed by the first development of those analog insulins, like Kevin and Kashif mentioned. And we flipped the script from twice daily NPH and regular to really trying to give insulin more physiologically with a long acting and then the short acting insulin with meals. But even the quote unquote rapid acting insulins that we inject, like Novolog and Humalog, are not fast enough to cover meal excursions of glucose like somebody who's got a normally functioning pancreas. So we just need better therapeutics. And one option is to figure out how to get insulin into the body in a rapid way. And probably one of the most rapid ways to get anything into the body is through the pulmonary vasculature, if you can get at that there. And so there has been the development of an inhaled insulin product. And it's not the first one. There was actually a product on the market before that just was unfortunately a commercial failure, but actually worked quite well. I think because it was just a different paradigm for people and because it was a bit clunkier to use. But now we have this inhaled insulin called Technosphere insulin. That's now FDA approved for adults. Hopefully we'll get a label in pediatric use. That is truly a rapid insulin. It's far faster than any injected insulin we have. And it's because the insulin's attached to these little microparticles that go in through the lungs. They dissociate at the pH of the lung, go right into the blood vessels, and are circulating within a couple of minutes as opposed to the 10 or 15 minutes that it takes subcutaneous insulin to get out. And it also clears faster than those subcutaneous insulins. So it's really much closer to the replacement of a normally functioning pancreas that we'd love to be able to provide for our patients. And so I'm excited about the future of inhaled insulin. It's got a small niche market, I think, right now in adults, but I think there's really a great opportunity to expand awareness about inhaled insulin and certainly see it used in the pediatric realm once we get that FDA label there.
I have a ton of questions. So when you mean inhaled, we have problems with the asthmatics, right? We have to put an aero chamber and a mask because the kids got to breathe in 10 times before they get down into the lungs. How does this work on a child who's 5 or 7?
Yeah, so we've been fortunate enough to be part of the clinical trials involved in inhaled insulin in pediatrics. And I've been impressed that kids as young as 4 and 5 can, in fact, inhale this insulin without any challenge at all. The device for inhaling Technosphere insulin, which is sold under the trade name of Freza, looks like a little pipe essentially. And there are cartridges that are color coded that you put in that tell you how many Freza units are in that cartridge. And the patients literally just put the tip of the pipe in their mouth and take a deep inhalation one time. And in that few microseconds, the drug is delivered to the lungs, and that's it. Patients are able to do this quite nicely. In fact, some patients take multiple inhalations for a meal, depending on just how much they're eating and how much carbohydrate they need to cover or how much correction they need to give. But it really gives the patients a lot of flexibility. It allows them to not be worried about late hypoglycemia because the insulin clears so quickly. And for most patients, it allows them to get even better coverage of their meals.
And does it need to be refrigerated or?
It does. It's still insulin. And so it is still a small peptide at the end of the day. And it's just like injected insulin, it should be kept in the refrigerator until it's going to be used. But then the cartridges can be taken out. It can be stable outside the refrigerator for quite a while, just like we talked about with pens or people on an insulin pump.
And then this is a misnomer, right? There is no human insulin, and nobody does blood transfusions or to get human insulin out and to give it to other patients. When people talked about humanolog, it's a genetically made insulin from E. coli. Is that correct?
Yeah, all the insulins we use now are recombinantly made. So they've taken the gene code for human insulin and inserted it into E. coli as a factory to make them pump out fully human insulin. So it is in that sense human insulin. That's how we generate it. And all the analogs are made by changing amino acids here and there to create different characterizations of the insulin when it goes into the subcu tissue and then has to try to find its way into the blood vessels. Some insulins, we want that to happen more rapidly. Others, we want it to happen more slowly. With the inhaled insulin, it is, again, just the human insulin gene that's being produced. And the reason we can use it to be so rapid is because of that unique delivery modality. So because it gets into the lung and directly into the blood vessels so quickly, it is exactly like the insulin that those of us who are fortunate to have pancreases that work make push insulin directly into our portal veins.
Well, and what would be the advantages? Just shows my ignorance in the field, but what would be the advantage if I have a child was already in a pump of using the inhaled insulin as opposed to the pump?
Yeah, that's the area that we haven't studied yet, but that I'm most excited about, I must say. Pumps and CGMs have been paradigm shifting for our field and are now considered a standard of care. And we certainly try to move the majority of our patients to those devices because can often get the best possible control with them. But they all still rely on these analog injectable insulins given subcutaneously that are frankly not fast enough to get good coverage of meals. And so I foresee, and we already are seeing, patients use inhaled insulin to augment their pump and AID systems because they know from their own experience that the inhaled insulin is going to cover a meal excursion far better than the pump can giving subcutaneous insulin. And so my hope is that we'll have commercially available systems whereby people can be on their automated insulin delivery system, but also have an option to use inhaled insulin for particular meals they know are challenging, or for particular times in their day that are challenging. They're going to play sports or they're going to take a nap or all those kinds of issues that may benefit from a different insulin. An insulin that has a different pharmacokinetic and pharmacodynamic profile. And so that's what the inhaled insulin offers.
And I know I'm asking you unfair questions because it's very early in the use of this delivery method, but do you think there will be a place for this product for type 2 diabetics where maybe they're in a GLP-1, but before they go out with their friends to eat pizza and beer, they inhale the insulin and it maintains a better hemoglobin A1C?
Yeah, I think anybody who needs insulin to achieve better glycemic control could be a patient that might benefit from inhaled insulin, depending on the situation they're in. We have given inhaled insulin to patients with type 2 diabetes, certainly in our most recent, even our pediatric study that hopefully will serve to get an FDA label for it. We intentionally enrolled both type 1 and type 2 patients. And although there were a few type 2 patients that were on basal bolus therapy, which was a requirement of that study, we did have folks who type 2 use it successfully as well.
And then you've also done a lot of studies. So before I go there, I was sharing a patient with Dr. Latif the other day when we talked, because from a primary care perspective, life is very frustrating. And it is, because you pray and hope that the super subspecialist does the work they need to do, because you really are not, I'm not going to take a kid off their seizure medicines. There's not, and I'm not going to change the management of a insulin dependent diabetic without the endocrinologist saying that's what I want done. I think it's foolish. But I had a 3-year-old in the exam room this past Monday. She was diagnosed before the age of 2. Father has type 1 diabetes since the age of 7. She's autobody positive. She's celiac positive. And she's there for a 3-year-old. And I asked the mom how she's doing. She's fine. And I go, did you take your cell phone out so we can see how she's doing? So she did. And the child's glucose was 350. And she's only been in range 7% out of the last 14 days. And I said, and she presented in DKA because she's too young to even think about screening. Well, you could have screened her, but she didn't tell anybody that the biological father had type 1. And I said, where's her pump? Oh, that was irritating her skin, so we took her off of it. And I couldn't get her to stay long enough to do ketones from the urine. And I just see the disaster coming, but what else am I supposed to do? I can admit her to the hospital. They won't let me. Put her in the emergency room. They'll laugh. What else do I do? She had an appointment 2 days later on Wednesday. So I faxed a note over to the Peds Endo. But when you see that, it's just so frustrating from a primary care perspective.
Yeah, type 1 diabetes is an incredibly challenging disease to live with, one that requires patients and families in this case, the mom, to act like pancreas 24/7 with no break. It's different from many other diagnoses where doctor just says, take this pill and come back and I'll decide based on your labs when we're going to change it. These patients have to decide how much insulin they're going to give multiple times a day, depending on the situation. So it's inherently different than a lot of other things and really hard. And you pointed out, like, not a pump is not for everybody. People do have challenges with wearing the technology. They have skin irritations. The pump sites get blocked or kinked. So it's just another example of why we need more options for folks to use, better insulins, different delivery methods. Not that a 2 or 3-year-old's going to be using inhaled insulin, but certainly that child gets a little older, that might be an important option for them. Certainly if they just can't continue to use a pump for whatever reason, and maybe even with a pump, inhaled insulin might be good.
Yeah, here the problem was the parental unit. As telling George, as I was going over the history with her, and her father has type 1. I'm like, yes. Oh. And then it was like watching the computer. I love Apple. So when the computer gets overloaded, you see the beach ball going round and round. That's what it was. I said, have you taken gluten out of her diet? She's like, why? I said, I'm sure the endocrinologist told you she tested positive for celiac disease. Oh, I don't know much about celiac disease, but you're not supposed to be giving her bread or pasta. She's, oh, I didn't know that. Like,
It's unfortunate that she didn't know. Yeah, there's a big communication gap and knowledge gap for lots of our patients and families.
yeah, all I can do as a primary care doctor is send you to the gastroenterologist and see if they can reinforce and teach her. Now, you've done some studies on trying to either prevent or delay the onset of insulin dependency or exogenous insulin dependency. We're all insulin dependent. So what strikes you about that? Where are we in that arena?
Yeah, that's where I spend most of my time actually, is in that arena of trying to do clinical trials to delay progression of disease. And that's what really got me most excited to be a diabetologist, and type 1 diabetologist specifically. In the 90s and early 2000s, we tried many things that were really safe, low risk, fairly easy to take, like oral insulin, trying to focus on antigen specific therapies to retrain the immune system. But largely those things all failed to provide benefit. As better immunotherapeutics came around, though, we started seeing some success. And over the last decade, we now have 10 different drugs that have shown the ability to at least slow the disease process down and preserve people's ability to make insulin for longer if they get these immunotherapies versus placebo in the clinical trials. And that's led to the FDA approval of the first drug to delay type 1 diabetes, which is tocilizumab. And that happened because of an incredible 10-year-long study that we completed in TrialNet, where we screened family members of folks with type 1 to identify kids who had antibodies, and found those who had antibodies and dysglycemia, and treated them with this monoclonal antibody to CD3. And we're able to show over a decade of follow-up that the patients who got treated had far a slower progression to disease than those who didn't get treated. 2 years on average. Newer data suggests maybe even 3 years. And because of that, the FDA gave the company a label, and we're now able to offer that drug not just as a research tool, but as a clinical therapeutic to people who are 8 years of age and older. And hopefully that age will drop lower with some data that's being generated right now to delay their progression of disease.
Well, and some people will say to me, what's the difference? 2, 3 years, you're still going to require insulin. Why even bother test screening and doing all of this? And then 2 years later, 3 years later, after the infusion, they're going to still require insulin.
That's a common question. So the first is just on the screening issues. And you told us about your 3-year-old in clinic. If that kid had been screened at a year of age and likely would have been antibody positive, the likelihood of that kid getting DKA goes from 40%, which is the general population rate, down to 2 or 3%, which is what we see in kids who've been screened. So it probably would have saved that kid a potentially life-threatening ICU admission. That's worth the price of admission on its own. But then if we have the ability to preserve beta cell function, even if it still means you're going to need to take insulin exogenously, we know patients who make even the tiniest amounts of insulin on their own decades and decades later after their diagnosis, those patients have a far lower risk of complication than patients who truly make no insulin at all. And so because we know that, we try to explain to patients and families that certainly the first goal is to delay their need for insulin. But if we can't do that, the next goal is to provide them with better glycemic control. And then the next goal after that is to provide them with the longest possible amount of time that they can make some insulin on their own, even these small amounts that they may not notice on a day-to-day basis, but that we know are providing some protection against all the micro and macrovascular complications of type 1.
And it does maintaining some, even minimal or even less than minimal C-peptide activity decrease the number of pathological lows?
It does. There's really nice data now that even when we use these ultra-sensitive C-peptide measures that go down to 0.01 nanomoles, just for reference, we use like 0.2 nanomoles as our usual threshold for being in a clinical trial with an immunotherapeutic. So even 10 times lower than that, we can differentiate patients who are above that low threshold and patients below that threshold. And the patients above it have less complications than the patients below it. And that's true for the severe hypoglycemia. It's true for retinopathy, nephropathy, cardiac disease. So preserving C-peptide, which is how we measure that beta cell function, really is critical.
And why do you have a theory of why that is?
Most likely it's because that small amount of insulin production is reducing their overall glycemic variability. Certainly the patients who are treated with these immunotherapies and have robust initial responses, that's what we see. We look at their CGM tracings and we see that they may not be completely normal, like somebody without diabetes, but their variability is far less than other patients of similar durations of diabetes. So what's likely happening is, even though it's still a challenging disease and these patients may feel like they're taking complete replacement insulin, there's a little bit of help going on in the background. And every little bit of help we can get may be reducing their risk for complications down the road.
So let me see if I learned a lesson. So one, the most important thing is to screen and screen early, because screening by itself can prevent DKA, which has a mortality rate higher than measles. It's 3 in 1,000 at a minimum. Some people in some areas of the world, maybe 1 in 100. It just depends on what zip code of the world you live in. 2, at some really good medical centers like Kansas City and Kentucky, if they know that you have type 1 diabetes, you may not even spend a day in the hospital. When you require insulin, they will teach you how to manage the child without an ICU or an hospital admission. 3, the parent is educated so they won't have to drink from the water hose the moment that the child's in DKA with cerebral edema and the PICU, and they're telling you, you're going to act as the child's pancreas for the rest of his life because he won't be able to manage. And 5, you probably preserve some beta cell function that we can measure as C-peptide. And that has a beneficial effect, not just acutely, but over the lifetime of this patient. And in addition to that, we can use monoclonal antibodies to delay the onset of insulin dependency, which also improve the function of the pancreas long-term, leading to possibly, we don't know yet, much better outcomes.
You nailed it. You're an excellent student. So I would say that you have a far more superior knowledge of type 1 than many out there practicing general medicine, general pediatrics. But that's the kind of stuff that we can teach through echo programs and awareness, and hopefully start to roll out screening programs, certainly for family members of type 1, like your 3-year-old. And there's no reason that kid shouldn't have been screened early, but even perhaps for the general population. And as we have better therapeutics, it will become a moral imperative. It will no longer be a question of could we, but it'll be we must, because we'll say that the outcomes that we can generate from screening and offering immunotherapeutics far outweigh the costs of screening patients for autoantibodies.
I have to keep it simple because I'm not a professor. So what I've learned is in actual practice, nobody does a lead screen. There's 10 questions. The parents want to fill it out. I don't have time to read it. I just order the lead. You're not really supposed to screen everybody with lipids. It's not. The U.S. Task Force on Preventive Services says it's not science, not evidence-based. they don't look at cost. It's just not evidence-based. You're supposed to ask two simple questions. Anyone in your family died before the age of 50 from cardiovascular disease? Do high lipids run in your family? And if they say yes, you should screen them as early as 2 years of age. What in reality happens is everybody's too busy to ask those questions. And when you ask them, you get a mother like I did on Monday where the little spinning ball turns on and she just doesn't know the answer to the question. And she gets frustrated, I get frustrated. So what people do is they draw lipids at 9 through 11 and see what's going on. And so I think although the strongest science right now is for the ones that have a first degree relative, which is 15 times risk of developing type 1, to implement this in busy clinics across the country, it's going to have to be everybody at 2 and 4 years of age, because you take all the friction away and then it just gets done.
Yeah, I agree. The rate of type 1 diabetes in the US is 1 in 300. So it's not a rare disease. And 90% or more of our newly diagnosed patients do not have a family history. Even though there's a strong genetic component of type 1, it's not a single gene that's inherited autosomally, dominantly, or recessively. So you're right. I think for this to really become something that everybody does, it needs to become part of standard practice. And just like you check a hemoglobin or check a lead level, you might also, from a separate finger stick that you're collecting at the same time, send off for type 1 diabetes autoantibodies. I think that's the hope, that we'll get to that in the not too distant future. We certainly already have the capacity to do that. We are doing that in a number of clinics around the US to be proof of concept that it can be done as part of busy primary care practice. And hopefully with help from Breakthrough Type 1 Diabetes and the type 1 community, we've submitted a letter recently to the preventative task force that makes these recommendations. If ultimately they do agree with that, then the insurers and payers will cover it, and it can become part of standard pediatric care. I think we'll be able to pick up all those kids early, or nearly all of them, and we can eliminate DKA and offer immunotherapeutics and really change the way we think about type 1.
You know what? From the general pediatrics point of view, I'm a general pediatrician. See a lot of patients. I think the screening is a good idea. Logistically, 2 and 4, I think that's where it's supposed to be because it's the highest amount of autoantibodies. But you're never going to get somebody to go to the lab with their 2-year-old to get blood work. That's the problem.
Yeah,
This theory was already proven with the lead screening. So they wanted to have lead screening at 10 months, at 2 years, at 3 years. Here's a script for it. A majority of the time, it was not completed. Patients just would not take their little kid. Unless the doctor's office is the one that draws the blood, not going to happen. They're not going to willingly go. When they came out with the lead machine in the office where you can do a finger stick, then you had 100% compliance. And that's why I think
it's got to be easy. It's got to be fast. We do now have dried blood spot kits to do autoantibody testing, and that's what I'm suggesting is the way forward. People aren't going to take their 2 or 4-year-old or 6-year-old for a venous draw for a disease they don't think is likely to happen, that they don't know anything about.
But from the practical point of view, the dried blood spots, good idea. Probably can be engaged. Okay. But in a general pediatric office, in general, pediatricians are under reimbursed or underpaid for services in general. The AP always puts out all these surveys and recommendations. You should do this, that, and the other thing. This is just another thing that we have to do in our long list of things that get encompassed into the WellCare visit. And you wonder why you have physician burnout. It's a great idea. I believe we should help children. We should make people better. And it's the right thing to do for humanity and for the future of children. But if all doctors get burnt out because they have all these little extra things to do, there's not going to be anybody to take care of these kids.
So I think that's a very important point, and I think that's why I'm so proud of this resolution that leadership has presented at that P. Not only are they urging that it be put into the periodicity schedule, that children be offered screening at 2 and 4 years of age, but they're also asking that a CPT code be created to reimburse the primary care clinicians for the work entailed in doing this test. Because as Dr. Heller pointed out, 299 times out of 300, the test is going to be negative. I still have to sign off of that test in the computer. That takes time. And that cannot be bundled into the well child payment, because I'm really doing public health. I'm trying to prevent bad outcomes and prevent extra expenditures for the health system. And my time has to be valued in that part of the equation. But I'm tremendously proud about that resolution. And hopefully our leaders at the AP will pass it. And along with the task force, we will have strong guidance from the AP and the task force that this is the way to go in the future. Unfortunately, in New York, the kit that's commercially available, the state of New York does not allow it to be used. So it's a big problem. I want to ask you, each one of you, how close do you think we are to the cure?
I can start off because I get that question almost every day since I focus on immune therapies in type 1. We're closer than we've been in the past, but it's impossible to put a timeline on discovery. I think the type 1 diabetes community has been sold the idea that the cure is just around the corner multiple times over the last 40 or 50 years. And that's done a disservice to the community and to research. We have to appreciate that pace of discovery is unpredictable and unfortunately is most often slow. But being optimistic, 10 years ago, we had no therapies that could really meaningfully augment this disease, and we now have almost 10. So I do think that we're going to get closer and closer to a cure every year that more and more patients participate in clinical trials and help us answer questions about which drugs could work. But will it be tomorrow or 5 years or 10 years or 20 years? I really think is impossible for anybody to objectively say.
Yeah,
But with all this screening and with what you guys are learning from the immunotherapy point of view, yes, it could be 20 years. Some things take forever, but it could also be in a year, because you're getting so many data points and you're getting so much more information than we have ever had. Or am I being too
I'll tell you definitively, it won't be within a year. We don't have the tools to do it, but there's no doubt that the screening is going to be a logarithmically accelerating thing for us. And the biggest reason we haven't made more progress is because patients don't participate in studies because they haven't been screened and don't know they're at risk. It's very hard to find patients who are stage 1 or stage 2. Now that teplizumab's approved and as we have more screening initiatives, hopefully we start identifying nearly all of the 60,000 kids who will get type 1 diabetes this year before they have any clinical disease. And a large proportion of them will participate in clinical trials that will help us answer those questions more quickly.
Great. All right, Dr. Latif, sorry, I interrupted you. What did you go?
I think it's a paradigm shift, and we are at the cusp of this issue to be addressed and tackled. And I agree with Mike. It's the research, the studies, participation, and everything needing to change over time and immune system. So there's so many variables that need to be studied, but I think that the movement has really started and is going to accelerate.
How about you, Dr. Kaiserman? Because you've seen it from all perspectives, from the university, private practice, and now sitting in the boardroom.
I just want to bring it back to the overall conversation today and thank my esteemed colleagues for their participation as well. I think one of the key things as we work towards whatever we ultimately call a cure, that we have many more options for people living with diabetes today. And over the last 30 years, we have seen really an explosion of new ways to monitor glucose levels, to deliver and administer insulin. All these things are important steps towards improving the lives of people living with diabetes as we continue to work towards ultimately that goal of having the eradication of the disease.
Thank you. Any other remarks anyone has?
No, again, to that point, I think with all the available therapies that are exploding, I'm really having a very positive response to inhaled insulin in addition to all the injectables that we have and the AID systems. And so the therapy side is evolving, monitoring side is evolving, and now we have this screening and hopefully changing the natural history of the disease side of things. So really exciting times.
Wonderful. All right. Thank you very much for your time. Really enjoyed this conversation, learned a ton, and I hope to see you somewhere sometime soon.
Thank you so much. Thank you.
Appreciate it. Nice to meet you guys.
Likewise. Thank you for listening. This has been a production of the Pediatric Lounge. On the show notes, you will find links to our co-host and other important notes, as well as a timetable of the topics discussed today. Don't forget to follow us on social media and subscribe to wherever you listen to your podcast. Leave us a great review, as it helps us greatly. In the meantime, we will see you next week, the Pediatric Lounge. The conversations are not intended as medical advice, and the opinions expressed are solely those of the host and the guest.